Then for students, then evaluations for students.
Yes. Good morning, everyone. My name is Ritik, and I am a student in WBSU under Dr. Reddy's lab. My thesis work title goes as follows: Identification of Anthracnose resistance associated genes and long non-coding RNAs in tomato using Solanum pennellii introgression lines. So coming to the importance of tomato. So basically, tomato is one of the majorly growing vegetable across the globe with production exceeding 180 million tons worldwide. And in the US economy alone, it contributes nearly about $3 billion. Apart from the economic perspective, it is also rich in certain vitamins such as vitamin A and C and certain micronutrients. But the production of tomato is severely affected by diseases, and one of the major fungal disease associated with the tomato is anthracnose. So the characteristic feature of anthracnose is it produces a dark necrotic sunken lesion on the ripened fruit, which degrades its quality and also the yield. So the molecular mechanism of anthracnose resistance in tomato are poorly understood. So that gives me this research opportunity to explore this molecular mechanism using using the long non-coding transcriptome elements such as long non-coding RNAs and microRNAs. So to get into this molecular mechanism behind the anthracnose resistance, I used introgression line. So basically, introgression line is a line or a plant which is almost similar to a cultivated variety. In our case, it is Solanum lycopersicum cultivar M82. But there is only a certain segment of genome from the wild parent. In this case, it is Solanum pennellii. And the main point to note here is it lacks lycopene. So it does not turn red and it remains like a little pale greenish tomato. The figure on the right is a basic illustration of introgression maps. Like a certain chromosome segment only contains a certain part of the wild donor parent, and rest is all the cultivated parent. So coming down to the second part in my research is exploring long non-coding RNAs and microRNAs and their relationship, how they fine-tune the expression of the targeted mRNA. So basically, just a brief, long non-coding RNAs are RNAs that are more than 200 nucleotide long and that does not code for protein, whereas miRNA are small 20 to 22 nucleotide long. These also don't code for proteins. Interesting fact here is that these LNC RNAs sometimes sequesters or sponges the miRNA, which may be beneficial for the targeted gene as it allows it to express more freely. So the objectives of my studies is evaluating disease resistance, identifying the susceptible and resistant introgression lines, and then doing the differentially expressed genes and the differentially expressed LNC-RNA analysis. And the final part is to investigate their interaction, basically the miRNA, LNC-RNA, and mRNA interaction and how they change the expression of the targeted gene. So coming to the methodology, I used 50 introgression lines obtained from CM Kirk Tomato Research 50, 50 introgression lines. And fruits are harvested at full maturity. After that, I inoculated them with 2 strains of Colletotrichum, the fungus that causes anthracnose. namely Colletotrichum cocoides and Colletotrichum nigrum. After the careful inoculation, which is calibrated to 1,000,000 conidia per ml, the fruits are inoculated for 6 days, and then the lesion diameter on fruits is measured using a caliper. And from that, we have selected few highly resistant and few highly susceptible lines. And from them, RNA isolation is done. One important thing during the directional RNA sequencing is we use the rRNA depletion method, as it removes the ribosomal RNA, which is a major chunk of the overall RNA of the transcriptome. And the rest RNAs, which I am focusing on, non-coding and miRNA, so we can capture them quite easily. Another reason is mostly LNC RNAs lack poly-A tail, so the poly-A selection won't be feasible to catch them all. So after the DEC analysis, we have identified certain DGs and DELs, and then going through the miRNA identification using the psRNA target prediction and construction of a competitive exogenous RNA network, the CE-RNA network, and then we identified a few results which I'll follow up in the next slides.
Because we're currently signing on another device. So just say, okay.
So this is the rRNA removal or the depletion method. So basically here it helps us to capture both coding and non-coding RNAs. And basically the main point here is to capture the LNCs as well as we can. So coming to the result section, before that, the figure on the left shows both the parent for the introgression lines, the wild one and the cultivable one. And you can clearly see that there is no lesion diameter whatsoever on the wild parent, the Solanum pennellii. On the other hand, the cultivable parent, cultivar M82, that is LA3475, shows a significant lesion development to both the strains of the fungus. And to the right is the mean lesion diameter of the introgression line for both the strains. So we have identified few lines which almost show full resistance, and majority of them show varying degree of susceptibility. So with the phenotypic evaluation, we identified a line, LA4093, that show very minimal lesion development in both the strains, comparing it to an susceptible line, 4091, which show a significant anthracnose damage after 6 days of inoculation. So LA4093 was selected as a highly resistant line. And further, we have gone through the analysis using the cultivable parent LA3475 and the resistant line LA4093. So coming to the result section, here you can see that the LA4093 has shown very low number of DEGs, and all of them are upregulated. Comparing that to the LA3475, the susceptible parent, that has shown more than 1,000 DEGs, and same with the differentially expressed LNC RNAs. And the same trend for 4093 also should basis itself in the LNC RNA. So from this, from my research, I can infer that the resistance in the LA4093 is a much more controlled response, and similar pattern or similar evidence is also been observed in a similar study that is done on rice with the infection of rice script virus, where the resistant genotype showed a fewer targeted DGs, it's a targeted and controlled approach towards infection rather than total disruption in the transcriptional machinery of the plant. These Venn diagram shows the unique and the common DGs and same unique and common LNC RNA between those two lines. So these are the top 10 unique DGs from the resistant line. And here we can see these highlighted in the red. These genes have shown significant fold change. And like the genes such as glucosidase, these are primarily these hydrolyze or break down the fungal cell wall, which is a fungal defense induced in plant, and also certain serine threonine protein kinases. So basically, these kinases, what they do is they recognize the pattern. Any foreign invasion or any biotic cells, they recognize the bacterial pattern or the fungal pattern, and they activate the further downstream signaling, such as calcium ion signaling and further receptors downstream the plant. These are the, this table shows the common DGs between the two lines. Here, the point to notice here is certain genes here show the contrasting regulation, that completely opposite regulation, like highly upregulated in resistant and considerably downregulated in the susceptible one. These genes such as pectin, esterase, these are essential to maintain the pectin composition in the cell. Also, this glutathione transferase, it's a enzyme that helps to counter the reactive oxidative stress and to reactive oxidative stress and maintain the plant homeostasis.
And GDS esterase also is there. This is important for QT.
Yes, sir. GDSL esterase is like the further slides.
Yes.
So coming to the gene ontology, as you can see, the upper figure, this one, is for LA 1493, which has shown approximately like 10 enriched pathways or the terms, and mainly they are dominated by carbon fixation, photosynthesis, or Rubisco activity. Basically, we can infer that the resistant line here maintain its normal metabolism whilst the infection is there. On the contrary to that, the susceptible one showed approximately like 48 enriched terms across all three gene ontology categories: its molecular function, biological process, and cellular components. And mainly enriched terms are like DNA binding transcription factors, signaling receptor activity, which suggests that the further downstream signal in the plant is effective and it tries to maintain its homeostasis during the infection. This plot here is the like the genes whose response to infection differ in the two lines, and we can see from the data here that they are mostly enriched in DNA binding transcription factor activity and certain kinase activity. Also, the signal transduction in response to stimulus. So, what we can say here is these terms, like these are the genes that can tell us why there is a differential expression or the differential activity among these two lines whilst having the common infection. Now coming to the tag pathway enrichment. So it is done for the susceptible line, and majorly the biosynthesis of nucleotide sugar is the most enriched one. Other than that is mainly the plant pathogen interaction, in which the number of genes and the FDR is high. So coming to these pathway maps, the left one is for the susceptible one, and the right one is the interaction, like the genes that differ, that basically differ among the response to the same stress in these two lines. So from the left image, we can see that. So these red boxes are nodes. Basically, these are like receptors, and the gene contributing to these receptors. These are not single genes. There can be a family of genes. Multiple genes can be there. So here we can see 7 nodes that are lit. So which suggests that I don't think I can see it appropriately, but it is like the cell wall, the two vertical lines, and these two, what we call the boxes, are the receptors, the surface receptors. So basically, this is a bacterial receptor that recognizes the bacterial elongation factor 18 sequence, and when it gets in, when it attaches to that, it further signals for the another pathways, mainly the calcium ion pathway. Another key receptor here is a fungal xylinase. Basically, it detects the presence or the invasion of fungus in the plant. So these, like the surface receptor, and this one is a calcium signaling receptor. These contribute to primary immunity, what we call PAMP, pathogen-associated molecular pattern recognition. And coming to these are ETI, basically effector-triggered immune response. So here there are 3 receptors that have been shown the considerable enrichment, namely PTO, the classic tomato kinase, and PTI6, which gets activated by the PTO, the PTO receptor, and one is RPS2. So basically, these 3 help plant to respond to hypersensitive, like the hypersensitive response, and helps the cell or the plant to maintain the cellular health and basically prevent cell death. Coming to the right figure, here we can see only the primary immunity triggering receptors are active. Then with the same CAM, the calcium ion one, and the basic fungus recognition factors. And there is a complete absence of effector triggered immune response, ETI, which has been the key case in the susceptible line. One more thing, both looking down to the bottom right, this is a KCS gene, which is a long fatty acid, like it forms a long fatty acid chain. And it also supports the cell turgidity and prevents cell death. So this one is like highlighted in both the cases. So these are the basic receptors. The PTI receptor, EFR, ELF18, LU, EIX1 and 2. These are the classical tomato receptors. And calcium, CAM, CML receptors are also there.
Plant receptors.
Yes, sir. So what we can say from this figure is that this susceptible line, like it overactivates or it hyperactivates its signaling. On the other hand, the resistant one maintains its metabolism rather than triggering a chaotic immunity response to the pathogen. So I already told that these nodes are not single genes. There can be family of genes. There multiple genes can be there. So this table here shows the different like the receptors on the dark red node, and various genes associated with these receptors, like they can be gene families. So the blank in the resistant one shows that its expression is not beyond the threshold, and the alternate expression in the susceptible shows that it overly triggers its receptors and genes to counter the infection. Mostly, these are susceptible specific. That means that only the susceptible parent express them to a level that is above the threshold, except for one, that is this KCS. I told you the long fatty acid chain forming gene. It has shown a resistance specific. Basically, it is much more expressed in the resistant and not significantly expressed in the susceptible one. And these are all the genes like the CAM receptor and the gene associated with them, and other receptors such as RPS2 and KCS. So, different part of my study is to find the LNC mRNA pairs. So, that is 6 of cis LNC mRNA pairs are identified, in which 18 are LNC of 35 are unique mRNA target, because one LNC can target multiple RNAs. 28 are concordant pairs. Basically, this means that the LNC and the associated mRNA, the gene, if LNC increase, like the whole chain of LNC is increased, the gene also increases. Like they both go in the same direction, basically. And 7 are discordant, the opposite of this. And while investigating about the LNC, these few LNCs stand out, and among them, LNC MSTRG 14675 is the most potent one, and assigning them or annotating them, mainly they are associated with cell wall remodeling and lipid obgesmonic acid defense, which I will cover in a bit. So this LNC RNA, which I am focusing on, it controls 6 genes into this window, that is 100 kb window. And out of these 6 genes, basically these 5 genes are of the family, like these are mainly associated with lipid biosynthesis, and the GDSL and acyl transferase, these like activate the defense signal, the lipid defense signal, and the concordant expression implies the coordinated local regulation, that is cis regulation, that this LNC is regulating these genes within a 100 kb window. Like these genes are within a 100 kb window of this LNC. So now, coming to this table, this is a basic table that showcases various miRNA, microRNAs, and then degrees, basically how many targets are there for this particular microRNA. And among these 10, the first one, the microRNA 5303, stand out clearly. And interesting thing is that there is actually a previous study on this MIR 5303, and it is noted that it negatively regulates the tomato disease resistance in the late blight of tomato through modulation of reactive oxygen species and hormonal signaling pathways. And these are other noticeable mRNAs. This table here shows the miRNA and their targeted genes of the long non-coding RNA and other attributes such as expectation, like expectation score and alignment. And that tells us about whether the miRNA will cleave the mRNA or it will hold its translational machine. Either it will cleave or it will not allow the mRNA to translate and code for a protein. So now coming to this cytoscape, this cytoscape is basically an interactive network between long non-coding RNA, microRNA, and mRNA. Here we can see this microRNA 5303. It is interacting with various LNC RNAs and certain genes also, which I have shown that it has shown the highest degree. Another important microRNA that we identified is 10532, which is also associated with the regulation of certain long non-coding RNA and certain genes. So the key defense-related genes that are being modulated by miRNA are SOLIC09G 008790, which is a SRSF protein kinase, basically a kind of kinase. And another one is SOLIC11G 016990, which is a protein tyrosine phosphatase. Basically, it's a phosphate group. And well, what my study holds for the future is functional validation of candidate genes and also LNC RNAs, such as 14657, that one I pointed out earlier, and identification of resistant associated markers for the breeding programs, basically, and whether we can see or compare the cross pathogen analysis, whether these, like, during a certain infection, whichever genes are upregulated or they changed expression, whether these are these same kind of genes are also there in other species that show the same kind of behavior under a biotic stress. In my case, it is the fungal infection. So to conclude this study, like the main points here are resistant genotype, that is the LL4093 line, has showed a controlled gene expression. You can see from the DEGs, it has only shown few DEGs as compared to the susceptible one. And the key defense-related genes such as kinases and phosphatases were identified and will further go down for functional validation. LNCs and miRNA play roles in gene regulation during infection. So basically, the non-coding part of the transcriptional machinery, these LNCs and miRNA, they can change the expression of certain genes by halting the expression of LNC or the mRNA. Basically, miRNA sequesters or binds and keeps the messenger RNA free. So this LNC, the cytoscape network shows a competitive endogenous RNA-based population in which the same thing, a particular miRNA can change the expression of mRNA depending on its interaction with the concerned LNC RNA. And broadly, these findings will support the breeding of these disease-resistant LNCs. Finally, I'd like to thank my professor, Dr. Umesh Reddy, Dr. Padma, and also Dr. Carlos for being there with me. And rest of you guys, you are always helpful. And that's it from my side. Questions?